Hello everyone, welcome to another lecture of drug delivery engineering and principles. We have been talking about gene therapy for the last couple of classes and we will continue that discussion, but before we go forward let us just quickly do a recap of what we have been talking about in the previous class. So, in the previous class we were discussing, as I said, gene therapy, which involves delivering gene or repairing gene. So, this could be delivering gene, this could be repairing gene, or this could be something else as well. So, the delivery of the gene could be in the form of just the whole cell itself that carries your gene of interest, and then if the cell is not autologous, that means that if the cell is not from your own body, then you have to devise your drug delivery vehicle in such a way that it is protected from the immune system, and we have again discussed quite a lot about this in previous classes. And then the other way that we were discussing in the last class was delivering directly the DNA and making sure that the DNA then goes to whatever site we want, and we discussed there are several barriers to that. So, delivery of gene without the cell itself, so just the gene itself, and there are several barriers both extracellular and intracellular. So, extracellular barriers could include degradation by any kind of nucleases that are present in your serum, how it is going to reach the target organ, how it is going to reach target cell, and then and of course clearance by the immune system, and then intracellular barrier included how it is going to go past and inside the cell, how it is going to most of the time, because this DNA is fairly large molecule as well as ionically charged, it has to be through endocytosis. So, how it is going to escape those endosomes, and then once it escapes endosome, how it is going to move around. So, diffusion in cytoplasm, and then finally how it is going to reach the nucleus. So, all of these barriers were there, and in the process of that we said that it will be easier if we use some kind of a carrier. So, we discussed two, viral and non-viral. Viral vectors, as the name suggests, is using the naturally occurring virus and modifying them to carry your own gene of interest, and we discussed several pros and cons with that, and then we discussed non-viral vectors, which are typically polymers and lipids, and we are in this case we are basically trying to mimic these viral strategies and remove the cons out of it while keeping the pros. But it is not that simple, and we discussed again several pros and cons for both of these strategies, and depending on what you want to do, you may choose one over the other. And then since this course is drug delivery and we are mainly talking about polymers and other biomaterial, so then we dive deep into what are the different polymers that are used for gene therapy. So, some of them we discussed were cationic polymers, and other class we discussed was just encapsulating within the polymer. So, they may or may not be cationic. So, examples of cationic polymers were PEI, poly-L-lysine. Encapsulation could be through PVA, PLGA and other polymers. So, that is what we had discussed in the last class. We will take this discussion forward in this class. So, let us look at some of the major ones that are used. So, I mentioned PEI. So, it is polyethylene imine. One of the most used polymer for gene delivery, and the reason for that is it is highly cationic, and so it solves two purposes. One, it forms complexes with DNA. So, at the right ratio you will get different size particles and you can choose what size you want, And then the second is that since it is so cationic and contains lots of primary, secondary and tertiary amines, it helps in endosomal escape. So, that is what we discussed. Here is just some examples. So, you can have a linear molecule of polyethylene amine. So, in this case here is your monomer which gets polymerized, and you can see that there are tertiary, in this case this is a secondary amine. You have primary amines. But as you can see from the structure, there are actually no tertiary amine. So, that is what the linear PI is, but you can polymerize it further to get branched PI, and now what you have is you have all kinds of amines. So, you have, you can see here you have primary amine, here you have secondary amine, and then in the structure you also have tertiary amine. And all of these amines, specifically the nitrogen, has these lone pairs, or I guess this lone pair is already being used with this hydrogen, but all of this can absorb H plus ion through interaction through these lone pairs. And what that means is, let us say if this is an endosome. So, if this is my endosome containing a particle with these polyethylene amine molecules, then what does that mean? That whatever is H plus that is available in this endosome, and which is in higher quantity than outside, because the pH of the endosome typically is around 6, 5 to 6. So, some of these polyethylene amine can absorb these H plus ions, causing the increase in pH. Now, the cell wants to maintain the pH to 5 or 6, so it is going to pump more H plus ions, and now as it is also pumping H plus ion, it has to pump in to maintain osmotic balance more sodium and chloride ions in there to make sure that the osmotic balance is there. So, now what you are doing is you are creating an osmotic gradient in here. So, that is going to lead to water going in, and now the size of this endosome is fixed by the amount of vesicles that are present, or amount of lipids that are present in the vesicle, and as more and more water will go, it will start to swell, and after a certain limit it will just burst. So, if you have these molecules in large quantities in the endosome which can absorb lots of H plus ion, you can have this endosomal escape, and we have already talked about this. This is also called proton sponge effect, if you guys remember. So, if you want endosomal escape, basically you want to make sure that your molecules or polymers or whatever your drug delivery vehicle is able to absorb lots and lots of H plus ions, and again amines, all kinds of amine, primary, secondary and tertiary, are extremely good at this. So, you can see even in this little small structure how many amines are being present here, which I am just circling most of them here, and hence this is act as a very good carrier in taking your DNA and then making sure that the DNA comes out. And now once it comes out, the DNA of course is interacting with all this negative charge on its structure to the positive charge on the structure of PEI, and once it comes out, the ionic concentration outside is high, and because the ionic concentration is high in the cytoplasm, this interaction between the positively charged polymer with the negatively charged DNA is not as strong, and this DNA can break off from the particle and then can try to diffuse around and reach nucleus. So, here is just pictorial representation of what is just said. So, instead of having a DNA molecule which is negatively charged, it is not going to go in the membrane, you have now formulated it in a particle formulation where all this DNA is making the particle, and along with the PI of course, and this can be then taken up through endocytosis. And once it goes there, the pH as it drops to 5, or as it starts to drop, this is going to keep on absorbing more and more H plus, causing more and more ionic balance ions going in and increasing the osmotic pressure, and eventually it is going to burst. However, one note of caution is most of these cationic polymers, if they are highly cationic, can also be toxic, and one of the reason for that is these since they are ionically charged and they are positively charged, we have already talked about that cell membranes are slightly negatively charged. So, they can then ionically interact with the cell membrane in huge quantities. So, what can happen is your PI polymer will start to interact with the cell membrane, thereby first of all disrupting the function of all these receptors that are there on the membrane, and secondly also causing instability in the lipid membrane layer, so may even lyse the cell. So, that is why it has to be taken into account that PI can be fairly toxic, and if you are going to use PI, you have to make sure you are using it at a concentration and using it in a formulation that is not actually killing off your target cell. So, then chitosan, which is a natural polymer, PI is a synthetic, is again also used for this application, and as you can see it also contains in this case primary and secondary amines. So, very similar concept, however, this chitosan is not as heavy in density of these amines and positively charged. So, they can still complex with the anionic DNA, but they are not as charged as your PI is. So, this is not as toxic, it is also a natural molecule. So, it is fairly compatible, and it is also non immunogenic, it does not really generate immune response in a body. So, one of the other property of the chitosan is also mucoadhesive. So, that means that if it is given through a route which is a mucosal delivery, and if you remember from our route of administration topic, you will remember that mucosal delivery can be of several types. This could be oral, this could be nasal. So, any route that will encounter mucus in the surrounding will be classified as mucosal delivery. So, you will see that all of these routes are feasible, and of course, inhalation will also have mucus, you will have vaginal, rectal. So, all of these are mucosal delivery routes. So, if you deliver this chitosan particle, what will happen is they will tend to bind to the mucus very well. So, it is mucoadhesive, that means that it binds to the mucus, and that could increase its residence time at the location. So, instead of just getting uptaken by random cells, these particles will stay there, and for depending on the application this could be good or this could not be good, but you get that control with chitosan. And again, like PI or any other cationic polymer, it is going to form these complexes with the DNA. So, these will form complex with DNA, and like most cationic polymers, this is going to result in a formation of particle. You can change their ratios to get different size particles, and it will be good to encapsulate your DNA. So, as I said, there are amine groups again attached to the surface because of this chitosan primary and secondary amines. So, you can then also attach potentially ligands to the cell that you are targeting. Maybe you want to target to a cell that is expressing an X receptor which is not present in, let us say, other cells. So, you can put a ligand for the X. So, let us say X cell is the ligand. So, then it has a more chance of going into these cells than other cells. So, you can get it to be more targeting. You can also do that with PEI. So, all of that is there. One thing to note is why are the complexes important. So, not only the endocytosis, but once you have this, let us say red is your polymer, and let me just pick another color, and let us say green is your DNA. So, it is forming a ionic complex like this, maybe the size of this is about 100 nanometer. So, if you have a complex like this, what will happen? So, now you injected it into the blood, and we were initially talking about one of the barrier is that the serum components such as nucleases will degrade it. So, now if you have a nuclease, it can't really access the DNA very well. So, the DNA is also protected while it is traversing in the serum. So, you have basically killed two birds at a time. You have made sure that the serum stability is high, and you have also then ensured that this is going to be taken up through endosomal pathway and can also escape those endosomes. So, another class that we are looking at is PAMAM dendrimers. So, PAMAM is a polymer, and again like the previous two examples, this is also containing amines, both tertiary, primary and secondary. So, as you can see here, so you have primary amines, you have secondary amines, and you have tertiary amines. So, by the same logic, this can result in quite a bit of proton sponge effect, and what is done is you make dendrimers out of these. So, if you remember, dendrimer is defined by the generation. So, you have G0, G1, G2, and what you are seeing is as you are increasing the generation, obviously the molecular weight of this dendrimer is increasing, and then the amount of terminal amines is also increasing. So, you have got almost close to 750 primary amines that are available. The diameter is still within the size range of about 100 nanometer that you want, and because you have these many tertiary amines, you can get it to complex with the DNA, you can conjugate it to the DNA. Obviously, all of these amines can be used for any kind of chemical conjugation such as EDC NHS or other pathways, and you can also put targeting ligands as we discussed in the previous slide through these amines. So, that is another class of polymer that is being used. So, just some example on this is from a paper. So, what you are seeing is these PAMAM dendrimers were formed as gene carriers, and they were mixed with some DNA and to form a complex, the dendrimer DNA complex, and this is just electron microscope image. So, this is an electron microscope image, and what you see here is when they were given to different mouse in through lung. So, what was done is so you have lungs, right. So, you have your mouse lungs, you take the mouse, you anesthetize it, you put a tube into the trachea. So, this is trachea, and you put a tube. So, it is called endotracheal tube, and once you put the tube, you can then use this tube to thread a syringe or needle through it and deliver whatever you want to deliver in a liquid formulation. So, you can then just push the liquid through this tube. Obviously, there is a limitation of how much you can push, because you can potentially drown the animal if you put too much. So, usually for a adult mouse you can put about 50 microliter of your liquid. So, what these authors have done is they have injected 50 microliter of the liquid containing these complexes of dendrimer and DNA into the mouse, and one of the things that they are delivering is a luciferase gene. So, this is a proof of concept. So, it is a proof of concept. So, they delivered this luciferase gene, and then they monitored the animal to see if they are getting any uptake of these gene complexes into the animal cells, and whether then its result in production of the luciferase protein, because luciferase produces light in presence of substrate. So, it is an enzyme. So, it is going to, and then they are basically measuring the amount of light that is getting produced in 10 seconds per organ. So, they see if they inject naked DNA, they do not really get anything, it is almost 0. If they inject it, if they mix the dendrimers and DNA at a certain ratio in both the right lobe and the left lobe. So, depending on where you are seeing it from the animal perspective. So, let us say this is right and left, and so what you see here is both lobes have quite a high amount of light signal that is being produced. That means that not only your particles have been able to go to the lung, they have been uptaken by the cell, they were able to endosomally escape the DNA was able to then translocate into the nucleus and then was able to produce the signal. So, quite all those barriers were taken care of. Obviously, through the route of delivery they took care of one of the barrier, which was how it is going to reach the target organ, because this was directly injected into the lung. So, it was reaching directly the target organ, and even the amount of ratio that you are mixing with also makes a big difference. So, this is basically is NP ratio, and what does NP ratio means? It means the amount of nitrogen present in the structure. So, amines could be NH2, it could be NH, or it could be N only. So, amount of amines divided by the phosphate, and where does the phosphate come from? The phosphate is from the DNA, because again the phosphate, every nucleotide has a phosphate. So, the amount of phosphate that is present is directly corresponding to the amount of DNA that is present. So, that ratio makes a difference as well, because you will get different stability, different sizes of these complexes, and that is going to change the pharmacokinetics of them, the uptake pathways and all those will change. But what you are seeing here is using these polymers they get much better efficiency than let us say just a naked DNA. So, here are some more novel. So, these were the ones that are widely used, then there are some other reports in the literature. So, in this case they have taken a polyphosphoester. So, in this case they have taken a phosphoester backbone. So, as you can see here is a phosphate group, and they have then modified it with an amine to get that negative charge or positive charge, and the one major reason they are doing that, as I said, most cationic polymers are toxic. So, now you can see, so they first performed experiment to see whether compared to different polymers how is their toxicity. So, you can see they have chosen 3 polymers, one is their polymer the polyphosphoester, another is PLL which is polyl lysine, and another is PAI which is polyethyleneamine we talked about in few slides ago. And you can see that as you increase the concentration of your polymer along with the cells, mammalian cells, you see that even at low concentration you almost have half the viability than what you started with, and this is percentage, and as you increase it more and more cells die up to a amount that even at 100 microgram almost all the cells are dead. So, this is something definitely you do not want, but in case of their polymer they do see even at high concentration the amount of viability is almost 100%. So, that is what then they started with. This is further confirmation in an animal setting. So, this is they are looking at what happens when they inject this polymer into an animal. So, you can see the tissue looks fairly healthy, it is well organized in a saline injected as you would expect, but if you are injecting PEI you see quite a bit toxicity, and then with their polymer it is much closer to saline than it is with the PEI. So, that is what they started with, and then they did experiments to see if they can increase the INF alpha concentration by delivery of INF alpha coding gene, and so they have measured this concentration in serum, and what they see is in the naive group which has not been given any treatment, even after several days, of course you do not expect any change in the INF alpha concentration. In the group that was given a naked DNA, so without any polymer, you do not really see much enhancement. So it is almost at day 14 and day 21 you do get some more signal, but it is still not as high, but when you give the complexes you see quite a bit enhancement in signal protein at day 14 and 21, even more than your naked DNA. So this is after the intramuscular injection, and they are measuring the concentration in the serum. So again, if this has to work, it has to go through all those barriers, and only then this protein can be produced and enhanced. So again, you can see that using polymers help in enhancement of the signal. But then one thing to note, of course, is even though it has low toxicity, the polyphosphoesters do not really work as much as PEI, but then at least you are not worrying about the safety issues with that. Okay, we will stop here and we will continue rest in the next class. Thank you.